Without further ado, I'd like to introduce Diego. Thank you.
Thank you. Well, first of all, thank you for inviting me today. It's a pleasure to be able to talk to the Rotary. I've first heard of the Rotary. It might have grown in Spain recently, but I first heard about it in California back in 1990 when I spent a year with my family at Caltech, which is a university in Pasadena, one of the small but important universities in the US. And I actually attended one of their trips. We went into the desert with them. So not a member, but a big follower and admirer. I also want to start by acknowledging that we're meeting in the land of the Ghana people. And I also want to acknowledge that the work that we are doing up in the Flinders is actually in land of the Adnyamathanha. So, which leads me to start the conversation or start this presentation with what most of you might have an idea of what they might be. What are fossils? So you probably have had a fossil, or you've heard of the term fossils, obviously. Most of them are just the remains of organisms. That's the standard one that we can relate to. So we've got bones from T-Rex in this case, or ammonites like that big shell, or we've got trilobites, the third one along, or in very special circumstances, even the soft parts of organisms are preserved. That's what you see on the right-hand side. That's Archaeopteryx. It's only got not the bones, which are normally, because they're already mineralized, have a better preservation potential, but also the feathers, which would in most 99.9% of the cases would just get decayed really early as the organism, just after the organism dies. So fossils, the body fossils are all of those types. And then we go on to other types of fossils like trace fossils and other evidences. Here we've got footprints from dinosaurs, which you would have seen in documentaries. In the other one, on the right-hand side. Oh, what's happening? Yeah. So the other ones are trace fossils. So the feeding evidences of things like those trilobites I showed you above. So we lost the screen, have you? Do we have a remote that we can re-engage it? No, that's mine. Okay, but also things like eggs. Empty eggshells are, yeah, you can watch it. You can check out that one at the back end of the room. And also things like coprolites, so fossilized species. So they're not common, but even those very soft things actually get preserved. And the one that we actually talk about often, fossil fuels, things like gas, coal, and oil are evidences of marine and land organisms from the past. Yay, we got it? Perfect. So now the other thing that I think is quite useful once we know what a fossil is actually looking at time in our planet. So we've got Da Vinci's famous drawing here. We've got the first, if you put out your arms like this, your right hand is basically pre-life. The planet's still accreting things from space, and it's too hot to have liquid water. It's all in vapor state. When the planet starts to cool down, we've got the first evidence of life really early after that, and we've got single-cell life all through here. So this is pre-oceans. And then we've got single-cell life, single-cell life, single-cell, 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 single-cells, till we get to your wristband of your watch, and that's when you start to get multi-celled, and that's the next step over. And then you've got the Phanerozoic, the time of complex life in the planet. So only the left hand of your whole arm span. And then if we close up on that, we've got the Ediacaran, which is around where your wristband is. And then you go on to the multicellular, clearly multicellular, with the Paleozoic first, the time of trilobites, this bit, then the time of dinosaurs, which are the two middle phalanxes, and then the time of mammals, which is the last bit. And humans, if I actually clip the tip of my middle finger nail, human history disappears from this whole story. Okay? So that gives you a grasp of where we are in time and how long geological time actually makes of the history of the planet. In a different presentation, we've got, as I said, a very early life, with a very early planet, too hot. Then as soon as we got oceans, we got evidence of life. But that is very simple. Remember, single-celled or filaments, microbial Earth. And then we've got what we call Snowball Earth, best represented and actually recognized first in Adelaide of the whole planet, the first time that these glaciations, major glaciations, 60 million years of age, the first one of the two, which is the time from the dinosaurs to us. So very massive times of the planet frozen over north to south pole, basically. And there's two of those. One from Marina rocks, the Marinoan, the second one, and the other one from Sturt Gorge, the older one of the two. So in Adelaide, we define these massive glaciations, and then we go to the blue earth we're so familiar with. So the Flinders Ranges World Heritage is trying to capture, which is what I'm going to be talking about in the next couple of slides, what we have in Australia that nowhere else in the planet has it. So that's why the Flinders Ranges are so important. One of the things that it's basically the backbone to this time of the planet is the Bratchen Gorge Geological Trail. You start on the right-hand side on that grid part here. Oopsie. Cursor. Nope. Do you see the cursor there? Darn. Okay, so on the right-hand side of the slide, you've got the green band and the pink bands. Those is this Snowball Earth planet, so planet frozen over, no multi-cell life. Then you've got the GSSP, that yellow star there, and that middle slide. That's the definition of the beginning of the geological period known as the Ediacaran, the only one that's been described in the last 120 years. And it wasn't described in any of the other countries that wanted it. The Russians wanted it, the Chinese wanted it, Canadians wanted it, Australia won. And we've got this geological period named the Ediacaran after South Australian Ediacaran Hills. So very important milestone there. The beginning of the Ediacaran is defined here for the whole planet. And then towards the top of that Ediacaran, here with the light blue, you've got the Ediacara Biota. The Ediacara Biota, although it happens in the Ediacaran, it's only the last 10, 15 million years of that very, very long time geological period. And then you jump into the Cambrian. By this time, as you can see on the left-hand side, those faunas, they've got elements that are known to us around us. So you've got the sponges, you've got the sea urchins and sea stars, you've got the arthropods with their jointed legs, you've got the mollusks with their spiral shells, etc. It's already present by this, but not during the Ediacaran. So it's a major change. So what do the organisms of the Ediacaran look like? Well, you can see the most iconic here. You've got Tribrachidium top left, then Sprigina, Parvancrina, and Dickinsonia. And these four elements were offered to the Australian public to choose which one we wanted as South Australians as our state fossil emblem. Does anybody here know, except, okay, yes. So which is the state fossil emblem? Sprigina, exactly. So Sprigina, named after Red Sprigg, who was the person who discovered the fossils at Ediacara Hills. So the Ediacara Biota, the Ediacaran Geological Period was thanks to Red Sprigg, and South Australia chose Sprigina, named after him, to be our state fossil emblem. Like we've got other emblems. We've got the leafy sea dragon as our marine emblem. We've got obviously the merry magpie as our state emblem, etc., etc. So this is what a idealized slab with all of those organisms together would look like. At the bottom, you've got that big frond, you've got Sprigina on the left-hand side, you've got a few of these Tribrachidiums and Inarias and a number of other things, but that's not what it looked like. This is a rock that's basically an external copy of what those organisms look like. This is what we would have seen if we were going snorkeling in the Flinders Ranges 555 million years ago. We're looking at organisms that are all soft-bodied, no shells at all. They haven't been invented, quote-unquote, yet. And basically, we've got things that are crawling on that seafloor. That seafloor, the reason why it's bluish-greenish is because this is a microbial mat. These are just filaments of cyanobacteria, like the muck that grows at the bottom of your pools through the winter. So basically, organisms are in touch with that surface. They're either feeding from it, in a few cases, otherwise just using it as a surface to lie on and absorb nutrients from the water. So why is South Australian fossils so important? They're the most diverse assemblage of Ediacaran fossils. We've got them in Russia, as I said, a few in China, a few in Canada, in the UK, etc. There are Ediacaran fossils in almost all the continents, but the best diversity, the most different species are found here in South Australia. But not only do we have the most species, we've got the largest Ediacaran, which is this one on display at the museum. It's more than a meter long. It's about that big. And it's wrinkled at the bottom, so it could have been a meter 20 centimeters long. We have also the smallest one. Here's a reconstruction on the right-hand side. It's the size of a grain of rice, but he was leaving this trail in the microbial mat because he was feeding from it. So we already have evidence of movement and active feeding from the microbial mat. We also have, and that's just about 2 millimeters long. We also have all the larger organisms, about 5 to 10 centimeters long, that were actively scratching the seafloor to feed, just like modern mollusks do, to feed from that cyanobacteria. If any of you have aquarium or aquaria at home, and you know that people put snails in them to clean the walls, that's because these snails are feeding, just like Kimberella was doing, from that microbial mat that it's just growing because of sunlight. And we also have some evidence that there might have been things already beginning to float in the water column. The rest are all sessile on that seafloor, some of them capable of moving, right? But except this one, which we named after Sir David Attenborough. So what did we think these things were? So the first and most obvious process when they were first discovered in the 40s is this have to be really early animals. Charles Darwin pointed out, wait a minute, one of his big problems was there's no life before the Cambrian that he knew of. Of course, this was 150 years ago. But so he said, well, life didn't come out of the blue. It's an evolutionary process, a gradual. And one of his conundrums, one of his doubts was, how could it appear out of nowhere? Well, now we know it's not out of nowhere. It actually has its roots in the Ediacaran, older than the Cambrian. So what did we think they were? Well, people obviously said, oh, they have to be animals. They're moving around. That's what animals do. They're all, they're swimming, they're crawling, they're drilling into the sediment. So that's where we put them first. Some of them look like sponges. Others were purportedly looking like jellyfish. Others like sea pants, not unlike the ones we see around us today. Some flatworms and even arthropods. However, in the 1990s, there was let's take a step back and see if we're actually looking at something that fits into the animal body plan. We are very special. We have particular genes that create our body, and those don't come out of the blue. What we were seeing with Ediacarans is that some of them have a growth pattern that is fractal by branching and branching and branching that you see in ferns, for instance, that is not present in animals, yet it was present in some of these Ediacaran forms. It's like, well, okay, that's not an animal. So that you have to open the mind to see, what if we're looking at things that are not animal? This is so back in the multicellular evolution that there could have been other experiments. And that's what was suggested by Salaher by saying, wait a minute, we might need to be a bit less shoehorning and more open about the interpretation of these organisms. So some of them are suggested to be part of this big group, the Bendobionta. So you would have 3 types, the ones we've got today, of multicellular organisms. Animals, we feed on other things and move. We got plants, which are photosynthetic, and then we got fungi, which feed on the other 2. Okay, they get all their energy from decaying organic matter. This would have been a 4th one, only present during the Ediacaran and becoming extinct afterwards, that had other types of creating a body. Not going to go into much detail, other that single drawing in the center. This looked like a sort of an air mattress construction. So you've got a tissue in the outside and then cells in the inside of those tubes. And recently we've looked at this, some of the most complex Ediacaran forms, and we see that they actually do fit into the tree of animal life. So we see a gradual, they're not all animals and they're not all something else. We're seeing a few things that don't fit into the animal tree, but also things that are on the way to become animals, but not quite there yet. As you know, evolution is a very slow process, and things don't change. I don't wake up, a human, and yesterday I was an Australopithecus, right? So things are gradual. You change very slowly, and what we're seeing here is that we've got steps going towards that animal, caraplate or regio or group, but not quite there yet. Okay, so this is where we're working at. Nilpina fossil site. 550 million years ago. These are the beds we're turning over. I'll show you how we do that in a minute. This is work from my colleague Jim Gayling at the South Australian Museum in collaboration with Mary Droser from University of California, Riverside since 2003. So we've been turning over. These are beds that were dug up in the last 15, 20 years, and they're now part of as I'll show you in a minute, of the new Nilpina Ediacara National Park that we just opened a month ago. So basically, the slopes are covered in fossils. That's why visitation to this site has to be under guide, because almost every rock you turn over not all of them, but 80% of those rocks will have fossils in them. And these fossils can fetch tens of thousands of dollars in the black market. They're illegal to extract from Australia, but we still need to protect our fossils for future generations. So these are some of the fossil beds. This is a one tree beds. And this is how we work. This is slides from the last couple of seasons. So that's how we find the beds on the top left-hand side. They're on the ground. We trace over the edges. We know that they're all in the outside, on the underside. So what happens with these fossils? These were organisms trapped alive, bless you, trapped alive when the sand smothered them. And that microbial mat actually created a death mask. So you've got a copy of the external part of the organism, not the inside, just the external part. And so because it was smothered, in order to see the fossil, you have to turn the bed over and you're looking at the underside. That's why we need to undo that. And so we extract the bed. You can see top right, the slab, dislodge it, extract it, and then move it to a new place where you put up your whole bed. Top left, we're actually logging the position of each specimen. Each of those labels represents a different specimen on that surface, and each of the colors represents a different species. So you can see in however many meters we've got there, square meters, that there's already a huge diversity of fossils in some of these beds. And some of them are totally new. Those top ones, the ones on the top right that I show you, those are new species never seen before by any human. And they came out in the last couple of years, okay? And they're big. Some of them are that big. So there's something very, there's a lot of information still to be brought out. And here in the bottom, you see what we've been excavating. So the first 2 years, 2019-21, then last year, and then this last year, we went there in May and got another 2.5 meters of this sea floors, basically photographs, right, of the 500 million year old sea floors. So this is a video we've produced for we got an ARC, Australian Research Council, discovery project to drill through the fossil beds, to make a hole and get the sample out of the fossil beds. And so basically we've been able to extract the first 65 meters of the fossiliferous beds so we can actually check their mineralogy. These are the core images. As you go deeper and deeper, 56 meters, 65, down to end of hole, 75 meters. And we've got 2 of those drill cores, and we'll be looking at those because this might be able to tell us what age those fossils are. We only know that they're about 555 because the Russians have similar species and dated at that age. So we still don't know the Australian ones. And this has been supported by the state government. The station was purchased by the previous government, and now we've opened the Nilpena Ediacara National Park with the new government. So this is great to see that there's a bipartisan support for the most special fossils that South Australia has ever seen. So we're developing a Flinders Ranges World Heritage, and we're about here. We're preparing the dossier, and that will be sent to Paris, and hopefully will be UNESCO. Just one thing, if you're interested in how this might apply to life in other planets, we know that Mars actually had a similar evolution for the first 1.5 billion years in its history. So we know that there could have been life there. It would have been cyanobacterial, just like here. So we've actually checked it was lost. That atmosphere was lost through the solar wind because it doesn't have a magnetic field. And the blue Mars turned to a red Mars. So we've actually sent rovers out there to check what the surface looks like. And these are Ediacaran beds, and we're actually producing a computer algorithm to identify the presence and the development of those beds. And we aim to apply that to the Martian beds. We've got the same sedimentary layers in Mars as we do in the Flinders Ranges, but we don't know if there were microbes there. It's going to take decades to get a human over there. But we can start knowing now if there were microbes already 3 billion years ago in the Martian environment. And with that, I want to say you should be proud of where you are, because we got the very best assemblage group of fossils representing the earliest complex life in South Australia. Thank you very much.
Thank you, Diego. I think that's the first talk I've seen when you covered about 100 million years a minute. So we've got a couple of minutes for Q&A. So has anybody got a question for Diego?
I get to start us off. This is a probably more like a fun question. So you have all these interesting names. So is there a kind of a principle there? And if the principle is you get to pick them if you discover them, what would you call one if you discover a new one? Or if you found one already that's new, what did you call it?
Okay, so there are very strict rules as how you name a new species. And I've named something like 35 new species around the world that with new genera and new families. And so there are 3 things you can do. You can never, ever name it after yourself. Never. Okay, you can get one named after you, and that's been my honor with some colleagues that named a species after me. But you name it after a feature in the organism, like Tribrachidium, 3 branches. That's, it's got 3 branches, then you name the genus Tribrachidium. You can name it after somebody, Spriggina, or Dickinsonia after Dickinson, Spriggina after Sprigg, or after a particular place. So we've named things from Kangaroo Island. We're going to name them Cartaensis because Carta is the Narrangeri name for the island of Kangaroo Island. So you can name it after any of this way, and you've got the freedom to name it whichever way you want. I've got friends who are so sick and tired of naming species, they've named it after musicians and actors and actresses and stuff like that. So it's a bit crazy, but you have the freedom to choose the name as long as it's not your own name. Yeah. Other questions?
Thanks very much. What a fascinating presentation.
Thank you.
What's the future in terms of the work that you're doing? You've drilled a lot of holes and they go to assess all of those.
Yep.
And does LiDAR play any part in terms of your research and work there?
Yes, good question. So, yes, we've drilled those 2 cores, and what we're going to be doing with those is search for 2 sources of information. One of them is the date, as I said before, zircons. So the little crystals that are produced during a volcanic eruption are trapped in ash layers, and you can actually know with a very small margin of error how long ago that volcanic eruption took place. So we can know if these fossils are 555 or they might be 5 million years younger or older. Whatever we find is going to be an amazing, because it gives us context to this Flinders Ranges world nomination. The other thing we want to check is the environment. We want to see for geochemical indices or proxies indications of how much oxygen was there in the ocean, how warm was it, was there a lot of organic matter, etc. And then the other things we want to do with LiDAR, for instance, will allow us to identify where the beds are. One of the problems is you've got subhorizontal beds, so they're tilted like that, and those of you that have done some geology, you will see that actual contour lines don't follow those in most cases. And so you have very complex, you've got channels eroding into other structures, pre-existing structures. So you've got, deep incisions made by canyons, submarine canyons. So we want to understand how are these beds relating to each other. The ones that I've just shown you are from 2.5 kilometers north of the original ones, the ones that you will see if you get to Ediacaran Geological National Park. So there's fossils almost everywhere, and we want to see the relationship between them. Are they younger, therefore higher in the stratigraphic section, or are they older? Are they deeper in that succession of sediments? So there's still, plus the biodiversity, the paleoecology, the environmental proxies, etc., etc. There's many hundreds of hours and probably generations of work still to be done in just this national park. And this extends to the Flinders Ranges, across the Flinders Ranges. You've got the top of Wilpinna. How many of you have been to Wilpinna? Did you know that if you've walked up, you have crossed the Ediacaran fossils? There are Ediacaran fossils all around the perimeter of Wilpinna, which is it's a syncline. It's a little cup, and the top, St. Mary's Peak, is actually Rawnsley quartzite, which is the same rock that produces these fossils. So they're all over the Flinders. The Hyson Range, the top of the Hyson, the crest of the Hyson Range, that's also Rawnsley quartzite. Again, fossil sites along them. Very hard to get to, but they're still there. Okay, so there's a lot of fossils all over the Flinders, and there's a lot of work still to be done.
Diego, thank you.
Pleasure.
I do sort of that I have had a bit of an involvement in what you're talking about, but I'd like to just say to everybody that the enthusiasm that you're seeing there is fantastic. I was fortunate enough last year to actually visit where Diego and company are working. There are people from right around the world. I think Stanford University, Mary.
Mm-hmm.
There's money coming in from NASA, etc. And because of Mars, what's going on in Adelaide, I don't think we know enough in Adelaide about some of the great things that are happening here. And you've done that today to say that, but I do recommend to everybody, if you do want to follow this up at some point in time, the opportunities are fantastic to actually go and see a huge slab of rock lifted up, which is showing the microbial bed with the fossils, and then it becomes a jigsaw, an absolute jigsaw, because it breaks up. And these wonderful students and professors and that are there putting together great jigsaws. And to be involved in that, I think all of us have that opportunity, and it'll be fantastic to see. I can only talk from personal experience. At university, I did biology and all of that. Later years, I never worked on it. But in later years, I've become more excited about the potential that we're seeing in this state. And it's there, and Diego is one of the great drivers of it. And please, if you get the opportunity, you will never be someone who's disappointed in what you're going to look at. Thank you.
Thank you, Jeff. Thank you. Thanks so much. May I mention that there is a foundation, the Flinders Ranges Ediacara Foundation, which supports some of the research we're doing out there. The rest is done with ARC linkages from the Australian government and NASA. But yes, there is a way for each of you to become involved in proceeding and advancing the knowledge from South Australia's Ediacaran fossils. So thank you very much.
Thank you. I just want to echo what was said before. The golden spike that Diego mentioned is very accessible. So a two-wheel drive vehicle will get you there. So go and have a look. So I'd like to thank Diego for a fabulous presentation. We'd like to present you with a certificate and a Rotary teddy bear. So here's your certificate of appreciation.
That's so cute.
And a recent fossil. So all monies raised today go directly to Rotary community and youth projects. Your presentation helps that make them possible. Thank you.
Thank you very much.
So could members please stand and we'll thank Diego.